police can't use the DNA at a crime scene to try to determine who was there and who might have committed criminal activity. Axiom 4, and I'm not going through all 10, but Axiom 4 states that by destroying the biological source of DNA, the forensic DNA sample, the 13 pairs of numbers, is reduced to identity, and therefore it has minimal invasiveness. And that is the way it should be done. The police should actually destroy your biological sample once they obtain it, justifiably, and use it only for means of identity and not for containing all the other intimate information in your genome. Axiom 5, people have an expectation of privacy of the information contents of their DNA regardless of where it is, whether it's abandoned or shed on their persons or medical records. In other words, if you throw out a cup in a garbage can, it's generally understood that you have given up the privacy of that cup. But we're arguing in this book that you haven't given up the privacy of the genetic information on that cup, which may have some of your saliva or some of your cells. That information is yours and shouldn't be open for anyone to access and discuss and report, etc. Police, we feel, do not have the right to analyze medical, paternity, or ancestry information on DNA without a warrant. If the DNA is at a crime scene, then they can surely analyze it for its identification purposes. Axiom 7, individuals detained or arrested for probable cause have a conditional, diminished expectation of privacy for those loci of their DNA that are used to establish their identity. But if the charges are dropped and they are proven innocent, they regain the full expectation of privacy. The DNA profile and its biological source should be destroyed. It shouldn't be retained. They have not committed any crime. So that's another one of the issues that we have dealt with in this book, Genetic Justice. There is a tension between the medical uses of our genetic information and the forensic uses. And the medical uses are getting more and more concerned about our personal privacy, and the forensic uses are getting less and less concerned, instead of there being a convergence between these two uses of our genetic information. Now let me give you just a story that will tell you how complex this can be, and there are a number of stories in the book that we tell, but the Gary Leiterman case was a story that was featured on CBS and one of the documentaries on CBS News. Leiterman was a person who was prescribed painkillers for a problem that he had with kidney stones. But he did not stop using the painkillers and instead figured out a way to get more of them when he was not prescribed them. He was a nurse and he was able to access the painkillers even though he had no prescriptions. So he eventually got in trouble with the police and he was charged with a violation of the drug laws. The judge, however, said to Mr. Leiterman that if he went to therapy and he wouldn't be convicted of a felony. So Leiterman went to a treatment program, which he successfully completed, and he voluntarily gave his DNA to the police, who put it on a database. Then the police at the same time were trying to solve cold cases, cases that may have occurred 20 years ago. Jane Mixer was a University of Michigan law student, a brilliant young student, who was murdered in 1969. It turned out, after 35 years, when they put Leiterman's DNA on the federal database, it matched DNA that was on Mixer's clothing. And Gary Leiterman, who was 62 years old at the time, never had any previous police record. The only time he was ever involved with the law was when he took the medicines that he wasn't supposed to be taking. He was arrested and eventually tried for murder, even though there was no physical evidence that he ever had any contact with the young woman. So it turned out that in the discovery of Leiterman's the trial, that his was not the only DNA that was found on the DNA evidence of the young woman who died in 1969. They also found another man's DNA. This man, however, was 4 years old at the time of the crime, so the police did not pursue that individual. One of the explanations for the fact that Leiterman's DNA got on the crime scene was that it was analyzed in the same laboratory that the crime scene DNA was analyzed, and that there was a good chance that the DNA of the crime scene was contaminated with Leiterman's DNA because they were analyzed in the same laboratory, possibly by the same technicians. Well, as a matter of fact, Leiterman is still in prison, and there are still many questions about contamination. Now, if Leiterman had not given his DNA to the police voluntarily, his DNA would not have appeared in the criminal laboratory. It would not have been analyzed in the laboratory, and it may not have been contaminating the crime scene. It's not the first time that DNA has contaminated laboratories. They had to close down laboratories in Boston, in Houston, Texas, in Chicago because of contamination. So you might argue, what difference does it make if I put my DNA on a national data bank? Well, the Leiterman case suggests to us that it could make a big difference. You may be an innocent person, but your DNA may contaminate a crime scene, and all of a sudden you're going to have to spend your life trying to defend yourself. So that's pretty much one of the stories that we tell in the book. There are many others about contamination and about protection of privacy with respect to forensic DNA. Thank you very much for listening to me. Something about the book. Okay. This book came about when I was on sabbatical in New York, and I was given a visiting scholar appointment at the American Civil Liberties Union. I met up with Tanya Simoncelli, who was a policy analyst for the American Civil Liberties Union, and she'd been working on privacy and working on DNA data banks. And I found some of the issues she was dealing with quite fascinating and challenging. Because on one hand, you have one of the most powerful technologies that criminal justice and police departments have seen in about 100 years since fingerprint testing had been developed. And everybody was quite excited by it. On the other hand, anytime you have a new technology, there's always going to be assets and liabilities. So we were trying to look at the balance of both the assets of forensic DNA testing, which everybody sees on television. You can't watch a crime scene show on television without DNA being part of that show. And it's usually something like, we've got your DNA, we got you. And, you know, and somebody knows that they got their DNA, and that's it. They don't have a defense anymore. In fact, part of what we did in this book is to show the fallibility of DNA as well as its appropriate and powerful uses. It's not infallible. So we worked for about 3 years trying to understand the different ways that forensic DNA is used. I spent part of my year in a forensic laboratory school in New York, watching students do forensic DNA analysis, so I had a fairly good sense of how it was done. I also went to some of the best laboratories in the country to see how the professionals do it. So it was quite a challenge, and we tried to come out with what we felt was a proper balance between personal privacy and the appropriate uses of this powerful technology to catch criminals who deserve to be caught. Yeah.
You know, you mentioned an interesting point, is there any way, something analogous to, say, isotopic analysis, that will allow you to determine the age of a DNA sample? So it seems to be essentially ageless.
That's a terrific question. Can you find out more from somebody's DNA than just their identity or their predispositions to disease? As a matter of fact, there's been some companies that developed that thought that they can actually, from your DNA, get a picture of what you look like. Now, that does seem a little strange, but yes, there were companies that were developed that were trying to create profiles of an individual from their DNA. I would say that the strongest evidence of a phenotype from DNA is red hair. If you've got red hair, there's a particular segment of your DNA that predicts pretty well, about 85% of the time, that you're a redhead. It doesn't work well with other colors of hair. But I don't know about age. I haven't seen anything about predicting age. But they certainly can predict certain diseases that you might have. If there's a certain segment of the DNA that predicts Gaucher's disease, for example, they can certainly predict at a pretty good predictor whether you have Gaucher's disease. But I haven't seen anything about age. But that's...
Yeah, actually, to rephrase, what I'm specifically thinking is there's some way that the samples try to be modified or something intrinsic that would allow someone to know roughly when a sample was taken so that cross-contamination with an older sample would be less likely.
I don't know of any way that they can tell you when the sample was taken. Unlike, for example, if they get samples of bacteria at a crime scene, they know what happens to the bacteria and how long it can last and when it died or something like that. And there is a time segment that they can tell you. Or if they see certain organisms at a crime scene, they can sometimes predict fairly well the time of death. But there's nothing like that I know with DNA. Now, in terms of the age of the individual who left the DNA, well, we're beginning to learn something about telomeres. These are the things that drop off in your DNA as you age. So it wouldn't surprise me that someday somebody might be able to correlate the number of telomeres that are left with the age of an individual. So I wouldn't rule it out. Yeah?
Except for contamination, are there other things that make a DNA sample not so reliably your sample?
Well, first of all, and I didn't get into the details of this, the only real way they can tell that it's your DNA is if they sequence the entire DNA of you and compared it to someone else. That's too expensive right now. So they use the 13 sites on your chromosome, and they have 26 letters. Now the real question is, what's the probability that somebody else out there would have those same 26 letters who wasn't